Hello everyone, welcome to my channel. I am going to discuss the biochemistry MCQ of Dr. MGR Medical University, Tamil Nadu. So let's start. So the first question is the marker enzyme of mitochondria is. So in the cell, different organelles have different markers such as endoplasmic reticulum has glucose 6-phosphatase as a markers, Golgi complex having galactosyl transferase as a marker, lysosomes acid phosphatase, mitochondrial inner membrane ATP synthase. So here the question was the marker of mitochondria. So the answer would be that is the ATP synthase, right? So the first answer is ATP synthase. Now the next question is metabolic function of endoplasmic reticulum includes all except, as you know, endoplasmic reticulum has a role in biosynthesis of protein due to the presence of ribosomes on the endoplasmic reticulum, right? Second, in the drug metabolism also they have a role, but degradation of protein is done by the lysosomes. So the here answer is degradation of protein, right? Last one is the ethanol oxidation. So ethanol oxidation has some tricky points because some of the alcohol is metabolized in the endoplasmic reticulum through a system which is known as MEOS, that is microsomal ethanol oxidizing system. So it is an alternate pathway of the ethanol metabolisms that occurs in the smooth endoplasmic reticulum, which converts ethanol to acetaldehyde, right? But in chronic alcoholic patients it occurs more, right? So here the answer is degradation of protein as it takes place in the lysosomes, right? Now the next is the third question. Examples of the absolute specificity of enzyme. So first of all we have to know what is the absolute specificity of enzyme. So the absolute specificity of enzymes means that one enzyme will catalyze only one reaction with the specific substrate, means one enzyme can act only on one substrate. Examples are lactase, glucokinase and the urease. Lactase breaks down the lactose, glucokinase phosphorylates glucose, and urease splits urea into the ammonia and the CO2. So you have to know all the three enzymes. In the next exams it can be asked as except also, that is which one of the following enzymes have absolute specificity except, right? So three examples you have to know: lactase, glucokinase and urease. So here the answer is urease, right? Now the next question is the mucopolysaccharides that serves as a lubricant in a joint cavity. It's a very easy question and it has the answer of hyaluronic acid. It is present in the joint capsules and it acts as a lubricant in the joint capsule, right? Now the Cory's disease is due to the deficiency of it. Is also a simple questions. These are the type of glycogen storage disease and you have to mug up these enzymes. So the Cory's disease is due to the deficiency of debranching enzyme, right? It is due to the deficiency of debranching enzyme. Okay, now the next question is the coenzymes needed for the pyruvate dehydrogenase complex include all except. This is another important question because pyruvate dehydrogenase converts pyruvate to acetyl-CoA, right? And the pyruvate dehydrogenase complex is consist of 3 enzymes and 5 cofactors, or you can say coenzymes. So 3 enzymes are, that is pyruvate dehydrogenase, dihydrolipoyl transacylase, dihydrolipoid dehydrogenase, right? 3 enzymes and the 5 cofactors are thymine pyrophosphate, lipomide, NAD, FAD and coenzyme A. So you can see the options here. The cofactors are, that is thymine pyrophosphate is there, coenzyme A is there, lipomide is there, but pyridoxine is not there. So there are 5 coenzymes, that is thymine pyrophosphate, lipomide, coenzyme A, FAD and the NAD, right? So the answer here is pyridoxine. Now the next question is, all of the following mucopolysaccharidosis presents with the mental retardation except. So as you know, mucopolysaccharidosis, Hurler syndrome, Hunter syndrome, Sky syndrome, Sly syndrome, Martiaux-Lamy syndromes, these are some syndromes name, but you have to know the mucopolysaccharidosis with no mental retardation, right? So no mental retardation is seen in Sky syndrome, Martiaux syndrome and Martiaux-Lamy syndrome, right? So here these three having no mental retardation, that is Ske's, Martiaux and Martiaux-Lamy. So here the answer is Ski syndrome, that it doesn't have any mental retardation. Now the next question, question number 8. In the well-fed state, all of the following changes occur in the adipose tissue except. So simple, that in the well-fed state, insulin is secreted and insulin will convert glucose into the glycogen, or any synthetic pathway that occurs is activated by the insulin. So you can easily guess the answer that lipogenesis is decreased. No, lipogenesis is increased, right? Simple answer, that is lipogenesis is increased because in well-fed condition lipid synthesis is accelerated due to the insulin. You can see the other options. Lipolysis is inhibited, right? Lipolysis would be inhibited. Hormone sensitive lipase is inhibited and the lipoprotein lipase is activated. These are all done by the insulin, but lipogenesis is increased by the insulin, not decreased by the insulin, right? Next, action of prostacyclins, right? So prostacyclins are basically icosanoids, right? These are derived from the arachidonic acids. So these are also known as PGI2, and from the name itself you can remember I means inhibition. Inhibition, what inhibition? It inhibits the platelet aggregation. So PGI2 has a function of inhibition of platelet aggregation, right? Now other functions are, it causes vasodilatations and cyclic AMP level increasement. So it has 3 functions. It causes vasodilatations, it causes inhibition of platelet aggregation, and it increases the level of cyclic AMP, but it doesn't decrease cyclic AMP level, it increases the cyclic AMP level, right? Okay, next question, the deficiency of beta glucosidase leads to, it is also an easy question, that is Gaucher's disease. These are the lysosomal storage diseases, and the Gaucher's disease is the most important disease, and Gaucher's disease is due to the deficiency of enzyme, that is beta glucosidase, right? So here the answer is Gaucher's disease, and Gaucher's disease is due to the deficiency of beta glucosidase. Other options, you can see that Niemann-Pick's disease. Niemann-Pick's disease is due to the deficiency of sphingomyelinase. Fabry disease is due to the deficiency of alpha galactosidase. Krabbe leukodystrophy is due to the galactocerebrosidase. So these you have to remember, right? Now the next question is, in the mitochondria MAO, that is monoamino oxidases, as you can see from the name, these are the enzymes which catalyzes the oxidation of monoamines such as serotonin, epinephrine, norepinephrine, and these are found in the outer mitochondrial membrane. Where? In the outer mitochondrial membrane. So answer is outer mitochondrial membrane, right? Next question, that is Crigler-Najjar syndrome. So the Crigler-Najjar syndrome is the basically an unconjugated hyperbilirubinemia, right? You can remember unconjugated hyperbilirubinemia by a code UGC. That is, unconjugated is seen in Gilbert's and the Crigler-Najjar syndrome. So in Crigler-Najjar syndrome, there is no conjugation of bilirubin with the glucuronic acid. If this is not conjugated, then bilirubin will not be excreted out from the urine, because bilirubin itself is insoluble in water. So it has to be conjugated with the glucuronic acid by the enzyme UDP glucuronyl transferase into monoglucuronide and the diglucuronide to make it soluble, and has to be excreted out from the urine. So in the Crigler-Najjar syndrome, where the conjugation is defect, the enzyme is defective, and the enzyme is UDP glucuronyl transferase. So answer is the severe deficiency of UDP glucuronyl transferase. Right. Next question. Serum alanine transferase level is elevated is simple in the any kinds of parenchymal liver diseases where the liver cells are damaged. This is a specific marker for parenchymal liver diseases. ALT is increased. Right. Now the next question. All of the following are the effects of insulin except. As you can see, insulin will always promote glucose conversion into the glycogen. So answer is simple, that it favors gluconeogenesis. It can never happen, right? Other options you can see, it has a function of lower blood glucose, right? Favors the glycogenesis, right? Favors the glycolysis because it increases the uptake of glucose into the cells and it occurs with proceedment of the glycolysis. So here answer is it favors the gluconeogenesis. It is not done by the insulin because question was except, right? Now the next question is, all of the following are the biological effect of insulin on the lipid metabolism. Same, if there is insulin, it has to be synthetic function. As you can see, first option, that is a stimulation of acetyl-CoA carboxylase. Acetyl-CoA carboxylase converts acetyl-CoA to malonyl-CoA in the fatty acid synthesis. So it will stimulate the acetyl-CoA carboxylase. Now the second is inhibits the HMG-CoA reductase. So HMG-CoA reductase is a limiting enzyme in the cholesterol synthesis pathway. So insulin will increase cholesterol synthesis, not inhibit. So the answer is inhibition of HMG-CoA reductase because it will increase the HMG-CoA reductase activity, not inhibit. Right. Third is inhibition of hormone sensitive lipase. It is done by insulin. And the last is a tricky one, that is stimulation of glycerol kinase because glycerol kinase converts glycerol along with ATP into the glycerol 3-phosphate. This glycerol 3-phosphate with the help of enzyme glycerol 3-phosphate dehydrogenase is getting converted into the dihydroxyacetone phosphate and it enters into the glycolysis. So insulin increases this pathway. That's why this is the function of insulin, that is stimulation of the glycerol kinase. Right. Next. Modifiable risk factor of serious diseases. So cardiovascular diseases has two types of risk factor. One is modifiable, another is non-modifiable. Non-modifiable risk factors such as age, sex, family history, this cannot be modified. But modifiable risk factors which are acquired risk factors such as smoking, cigarette smoking, obesity, diabetes, these are the modifiable risk factors. So here they are the modifiable risk factor is cigarette smoking. Right. Now the next question, Barlow disease is due to the deficiency of. So the Barlow disease is basically infantile scurvy, and scurvy, as you know, is due to the deficiency of vitamin C, right? All are the sources of vitamin E except. So vitamin E is found in all the vegetable oils such as wheat germ oil, sunflower oil, safflower oil and the cotton seed oil. So if you have any non-vegetarian sources, then it has to be low in the vitamin E content such as fish liver oil, right? So any vegetable oils having the rich in the vitamin E, right? Now the next question, vitamins needed for the transamination reaction. So the transamination is basically transfer of amino group from one amino acid to the keto acids and forming the corresponding amino acids and the keto acid. And in this transamination reaction, you can see there one cofactor is used on, that is pyridoxal phosphate, right? So the pyridoxal phosphate is the coenzyme or cofactor used in the transamination reaction, right? The last question, all of the following causes the hypokalemia except, you can exclude this question, such as one option is hemolysis. In hemolysis or in the RBC destruction, you can see easily that the intracellular potassium is getting outside, so the potassium level will be increased due to the RBC destruction. So here answer is simply that is the increase in hemolysis, there is hyperkalemia, right? That is hyperkalemia. Rest in all conditions there is hypokalemia, such as Cushing syndrome, renal tubular acidosis, and adrenal genital syndrome, right? If you have any query, you can ask or you can post it in comment box, or if you have any confusion regarding the answers, or if I have made something wrong in making any answer, then you can also comment in the comment box. Thank you. Hello everyone.